Hello everyone, welcome back to my channel Pari's tutorial. I'm back with one more interesting video. So let's get into the class. So in the last class, I talked about the DNA replication, the enzymes and proteins involved in DNA replication, and how the experiments were being proved regarding the semi-conservative mode, right? So now it's time for us to discuss about the biomolecule called RNA, right? So we have already gone through the structure of ribonucleic acid already in the last class. Today I want to concentrate on the types of RNA, because in the next class, after from some classes, I might start discussing about the protein synthesis that happens in the cell, because based on it, before that we have to have a clear idea about transcription, translation. For that we should have the clear idea about the RNA, what are the types of RNA, right? So RNA is considered to be one of the genetic materials previously, but DNA is having more stability than RNA. So RNA is still considered to be genetic material for some viruses, right? Okay, so sorry. So what are the types of RNA? So there are three different types of RNA. They are mRNA, tRNA, and rRNA. So this RNA acts as a crucial link between genetic material DNA and proteins. Okay. So what is mRNA? mRNA is a messenger RNA. Okay. So when you take a DNA example, like if you got a 5 prime side and a 3 prime side, right? Its complementary is like 3 prime and 5 prime. Okay. So what happens in mRNA transcription is, the mRNA is a single stranded nucleotide, which comes from a template, which where DNA acts as a template for production of mRNA. Okay. So for example, DNA got two strands, like it's a sense strand, this is an antisense strand. Based on this antisense strand, this particular antisense strand would act as a template for the production of mRNA. So we can simply say mRNA is a sense RNA that is produced from the antisense strand of a DNA. This antisense strand of the DNA would act as a template. Okay, so that is what you call mRNA. So it is a single stranded base complementary copy of three antisense strands of transcription unit. Okay, so you have already gone through what is eukaryotes and prokaryotes, right? Prokaryotes are having a cell doesn't have any nuclear membrane, and it doesn't have any proper nucleus formed, nucleolus formed inside it, right? So eukaryotes, these are all the cells who have which have a perfect nuclear membrane, everything, right? So the mRNA is different in different eukaryotes and prokaryotes. In prokaryotes, translation proceeds along with transcription. So what happens in prokaryotes? There is no proper nuclear membrane. There is no proper nuclear membrane, right? So due to which the transcription, which is already happening in nucleus, where there only the translation also, the protein synthesis also would happen in prokaryotes. But in eukaryotes, what really happens is, you got a perfect nuclear membrane surrounding the nucleus, right? Once the transcription happens in nucleus, that particular mRNA would come out of that nucleus, which is ready for the translation of proteins. So all the things like splicing, addition of polynucleotides, and what you say, removal of the introns and all, this is called the when you are making the pre-mRNA, right? So before it enters into the translation part, it has to be ready. So for that, there are some changes that would happen for mRNA in eukaryotes. So that all would happen in nucleus. So once that is ready, the mRNA is ready for further translation. That particular mRNA would now move from the nucleus to the cytoplasm of the cell, where the ribosomes are present. So that is how in eukaryotes, transcription and RNA processing, that's what I'm talking about. RNA processing has got many changes. That particular eukaryotic mRNA needs to have addition and subtraction things, and that I will let you know. So and then that mRNA is ready for your translation. So once it is done in the nucleus, they immediately get transported to cytoplasm, where translation or the protein synthesis takes place. So what is mRNA in polycistronic? Yeah, you should be hearing the terms like polycistronic and you say monocistronic. So what are these polycistronic and monocistronic? For your information, I'm simply drawing a small diagram so that it would be easy for you to understand. Okay, see, for example, let me talk about my prokaryotic mRNA. Okay, so it has got a 5' side and a 3' side. So what happens in mRNA of prokaryotes is it has got many cistronic regions. That means one single mRNA will be coding for more than one protein. That is termed as polycistronic mRNA. That is why it has got, for example, I'm talking, okay, these are the things. You have got a leader sequence here. You have got a trailer sequence here. I will tell you what are those. Okay, so this is your cistron, and again this is your another cistron where the translation occurs, and this particular part is called intercistronic region. Okay, this is called intercistronic region, and that obviously won't get translated. So this part, the leader part, won't get translated, and the trailer part won't get translated. Only the cistronic part, as mRNA in prokaryotes has got many cistrons in it that would code for many polypeptides, right? So it is called polycistronic mRNA. In the same way, let's talk about the eukaryotic mRNA. So eukaryotic mRNA has got a cap here and a polytail here. I will tell you what are those polytails and all. It's a tail sequence, all 8 tails, okay, and it has got a trailer, obviously, which don't get translated either in eukaryotes also. Okay, this is your leader sequence from where your translation would start. So this part is what you call cistron. This complete cistron codes for only protein coded by one cistron, right? That the proteins that is coded by one cistron is only, it is called monocistronic. It doesn't have any intercistronic regions or any other extra cistrons. Okay, that is what you call the eukaryotic mRNA is polycistronic, and prokaryotic mRNA is polycistronic, whereas eukaryotic mRNA is monocistronic. Okay, now let's go for the slides. See, what are the initiation codons? See, for every mRNA molecule, for the protein translation to start, it has got a AUG site, right? See, what is this? AUG is the initiator codon. It could be UGG also. Apart from that, this is the 80% mRNAs have AUG as an initiate codon. Okay, and this goes for formyl methionine tRNA. Okay, the tRNA that brings the codon for complementary codon for this AUG is formyl methionine tRNA. Okay, the tRNA, would you know that, right? So for mRNA, once the mRNA is arrangement of the ribosomes and tRNA, mRNA. Okay, I will talk to you later in molecular biology in detail. So as of now, I thought these are the initiator codons that I want to discuss, and at the same time, you got a termination site even in mRNA. What are those terminate sites? I will tell you. See, there are 3 nonsense codons. So codons which does not code for any protein. They are UAA, UAG, and UGA. UAA is for amber, US is for ochre, and for opal. Okay, these are the 3 nonsense codons. Okay, these nonsense codons, either of them, like it need not be all the three at the same position. Some mRNAs would have this as their termination part, some mRNAs would have this as their termination part. Okay, so if the tRNA recognizes this particular site in mRNA, then it stops the attachment of tRNA to the ribosomal part, and hence the translation is completely stopped, and you will get your polynucleotide chain, like polypeptide chain completely after the translation, right? So these are the nonsense codons. Okay, so this is how in brief I am telling you how the arrangement of mRNA would be in both eukaryotes and prokaryotes. Okay, now see, I told you, so chain termination site has any of these nonsense codons like UAA, UAG and UGA. So next go for the next site. What is that? Right. So I told you the prokaryotic mRNA has got a half-life of only 2 minutes. That they are called short-lived mRNA. They may ask you a question which is short-lived, either eukaryotic or prokaryote. Okay. Prokaryotic mRNA is having a half-life of 2 minutes. Eukaryotic mRNA has a half-life of 6 hours. Okay. And the maximum mRNA half-life is seen in plant seeds, which is about 24 hours to 4 days. Okay. So it's just for your information that I am telling you. Okay. So what really happens in translation initiation prokaryotes? So this is a basic thing I am touching, but in molecular biology I will explain you everything in detail. So here, translation initiation in prokaryotes, not only AUG, I told you it's not only the AUG site initiator codon, you have UG, GUG and UUG also for your translation initiation at near your 5 prime side cap, right? So the 5 prime leader of the bacterial mRNA has a consensus hexamer sequence. Yes, here I want to talk to you. What is Shine-Dalgarno sequence? This is the Shine-Dalgarno sequence. What happens is, this particular sequence is present in, if this is the mRNA that I am talking about, okay, and this is your initiator codon which has got UAUG, right? So it is located 7 base pairs upstream. So this part is called upstream and this part is called downstream. Okay, like before the AUG itself, you got a sequence called Shine-Dalgarno sequence at the 5 prime side of your bacterial mRNA. Okay, this particular Shine-Dalgarno sequence is a highly conserved sequence, right? What happens is, in bacterial ribosomes, you have a subunit called 16S rRNA, right? I will tell you in the next slide itself what are the subunits of prokaryotic ribosomes and eukaryotic ribosomes. Okay, that particular 16S rRNA would have a sequence which is same with the Shine-Dalgarno sequence of your prokaryotic mRNA. What happens is that particular rRNA with that sequence would bind here. The ribosomal part of that rRNA, 16S rRNA, would bind to this particular sequence so that the ribosome, so that the translation would start. So this is what the Shine-Dalgarno sequence is, and it is very conserved hexamer sequence. See, so this Shine-Dalgarno sequence has got complementary base. That's what I'm talking you. How the RNA would bind to that is mainly based on their sequence. So this got a sequence where immediately the complementary base like 16S rRNA would bind there. Okay, so it is in the 3 prime direction, obviously. So if this is in the 5 prime direction with Shine-Dalgarno, and 3 prime would be here with the correct sequence. Okay, this is the mRNA sequence. This is allows the smaller subunit of ribosomes to bind to the mRNA during translation. So that is how the ribosomes move, and the mRNA would go for the for the chain proceeding. Okay, so next, this is what I am talking about, translation initiation site for eukaryotes. Till now we have discussed about in prokaryotes, all everything, right? Now let's start about translation initiation site for eukaryotes. In eukaryotes, you already know that. See, this I want to talk about. This, let's say, okay. So what I want to talk here about is see, the prokaryotic ribosomes are 70S and the eukaryotic ribosomes are ATS, right? So what is S? It's a Svedberg's unit. How you define the eukaryotes, okay? What are the subunits here? These are the subunits of sorry, these are the subunits of ribosomes, okay? So this is the 70s and this is the 80s. So here what I want to talk to you is, see, in eukaryotes, the smallest subunit 40s of ribosome is bind to the 5' cap. So I told you in mRNA you got a 5' cap and a poly A tail at the back, right? This particular 40s ribosomes would bind to the 5' side of your mRNA. In some mRNA, the initiator codon lies within the 40 bases. Okay. In such cases, the 40th subunit moves from 5' cap till it reaches the AUG. I told you this is the AUG initiator codon, and this particular 40th ribosome would start here and it would finally move towards your initiator codon, right? This is how the translation would happen in eukaryotes. In the same way, let's talk about the termination side. In the termination side, I told you it's all does not code for anything. It could be either of it. It could be UAA, UAG or UGA. Once the ribosome reaches to that particular part, there are no other tRNAs entering to get that particular code, because you don't get, there are no the ribosome is 200 angstroms of diameter and rRNA associated with proteins. So ribosomes are composed of rRNA and proteins. Yes. So as I already told you, the ribosome in prokaryotes is 70s size, out of which 60% codes for ribosomal RNA and 40% codes for the proteins. So these are the subunits, 30s and 50s. In the same way, the ribosomes in eukaryotes are which are cytoplasmic. Why I have written cytoplasmic is because you know that there are other organelles in eukaryotes, rough endoplasmic reticulum and smooth endoplasmic reticulum. This I will cover in cell biology. Okay, the rough endoplasmic reticulum has got the ribosomes on endoplasmic reticulum organelle also. So based on it, I am telling you the ribosomes that are present only in cytoplasm. Okay, so ATS size. The ATS size would have 60% of the protein and 40% of rRNA. And what are the subunits of ATS? They are 40S and 60S. Okay. Now, this important functional site on ribosomes. The ribosomes of eukaryotes or prokaryotes is generally asymmetrical in nature. Okay. The two units are generally asymmetrically placed with each other. Okay. The two subunits only dissociate under low magnesium concentration. So if you want to dissociate, you have to use low magnesium. Okay. So larger subunit of ribosome serves, see, that's what there are two subunits, right? They are arranged in a model like this, where one particular subunit of ribosome would stabilize your amino acid tRNA, because it has to get invite the tRNA with particular amino acid bound to it, right? So that would be stabilized by one of the subunits of ribosome. The other part of the ribosomal unit, other smaller subunit of the ribosome would be stabilizing the mRNA. One will be taking of the mRNA, other would be taking care of the tRNA, right? That is how the whole functional site would be in ribosomes. So what are the two adjustment sites for amino acid tRNA? So I told you the ribosome has got two sites. They are site A and site P. So site P is meant for the formation of the peptide bond between the amino acids. This is the picture of the particular functional site of ribosome. Okay, I thought it is one of the old books, but I thought it would be easy for me to explain you better. See, for example, this is your mRNA, okay, and it is I told you, this is the 50th subunit. This is the eukaryotic cell, because sorry, it's a prokaryotic cell, because it has got 50 subunit here and 30th subunit. So it's a ribosomal functional site for prokaryotic organisms. Okay, so you got mRNA here, you got a P site here, you got a site here. This is a peptidal site and amino acid tRNA. This exit site, would stop your complete translation process. Sorry. Okay, this particular process. So these are the important points that I thought of covering today. So based on it, you have also one more thing that I want to clear. Okay, what are the subunits of ribosomes and what are the rRNA and how many proteins have been coded here? Eukaryotic ribosomes. So which are obviously ATS ribosomes. So you got 60S and 40S as their subunits. Okay. So what really happens in eukaryotes? So 60S, And I told you now let's talk about 60S. The 60S got 28S rRNA and it has got a 5.8S RNA and it has got a 5S rRNA. So these are the subunits of subunits of your 60S. So and these all would be associated with 49 different proteins. Okay, this all will associated with 45. In the same way, now let's start with the smaller subunit of eukaryotic ribosomes, nothing but 40S. So what are the subunits of 40S? It has only got 18S rRNA, which codes for 33 different proteins. So they may ribosomes. So you all know that prokaryotic ribosomes have got it's a 70S, 50S and 30S. Okay, so what are the subunits of the prokaryotic ribosomes? Now let's start with the 50S being the biggest subunit. Okay, so it has got 23S rRNA and 5S rRNA, which all code for 31 different proteins. Hope you got an idea what are the subunits, subunits of these ribosomes. So the other smaller subunit is 30S, and 30S has got a subunit of 16S rRNA. Okay, which codes for about 21 different proteins. So this was I am talking about the child-Dalgarno sequence to which it is binded. They may ask you what is which part of rRNA would be binding, which subunit of rRNA would be binding to the child-Dalgarno sequence. If example, if this is your child-Dalgarno sequence and this is your leader part, or it's called initiation part. Okay, let's start. This is your initiator codon. So upstream of this initiator codon, you got this Shine-Dalgarno sequence. To this Shine-Dalgarno sequence, this 16S rRNA would be having a complementary base, so that it would be easy for you for the complementary. Once they are complementary, the particular subunit of ribosomes would easily get attached and the translation moves. So the 30S subunit would be attached to Shine-Dalgarno sequence because it contains 16S rRNA in it. So this is the brief thing that I would like to cover in this class. Hope we have gone through rRNA and mRNA. Now let's go for the tRNA. So what is tRNA? So tRNA is a transfer RNA, and the most approved model is clover leaf model. It has got 76 nucleotides in it. Okay, so what is a model of this? See, this I want to explain it as simple as possible. So this is your tRNA model, and it has got all, I think 80% of the tRNA is double helical, because see it is almost paired, right? So it has only got a few loops in it. So 1, 2 and 3, right, and have 4 loops. Yeah, see this is called a DHU, dihydrouridine loop. Okay, and this is your 5' side, this is your 3' side, this is your acceptor region. Okay, and what happens is this particular anticodon, right, based on the mRNA, here the mRNA would be there. Okay, I will tell you detail in molecular biology. Okay, this particular tRNA with the complementary base of the mRNA would be base pairing here with mRNA. Okay, and based on it you will get your, what is the function of this dihydrouridine? It would help you to recognize the amino acyl tRNA, amino acyl mRNA. Okay, amino acyl mRNA has been recognized by THU, whereas this particular loop would be helping you for stabilizing your ribosome, right? This helps to locate or bind to the ribosome, and this particular thing would be used for the recognition of your amino acid transferase. Okay, to which you will form an amino acid formation and peptide bond and a chain of amino acids. So hope you got all the types of RNAs. You also got a diagram of how the whole thing happens. Okay. So apart from the RNA, all the types of RNAs, you also got an RNA called enzymatic RNA, which are called ribozymes. These molecules of RNA that has the ability to catalyze a trans-esterification. This is an important process that would be helpful for splicing, ligation, cleavage, and phosphate action. Okay, so as of now, I've covered the basic functions of rRNA, what basic functions of mRNA. Next class, hope you like the class. Please subscribe, like, and comment about this class in the comment section. Thank you and have a nice day.